Okay, good morning everyone. Slow. Welcome to lecture 24 of BMS 300. Okay, so today we're going to do part 4 of the nervous system, and I'd like to begin with an overview of what we'll be discussing. Alright, so we're going to start by talking about post-synaptic and also pre-synaptic regulation. On Monday's lecture we talked about what's happening at the synapse, that's the junction between the two neurons, and we see how the signal from the presynaptic neuron can be passed on to the postsynaptic neuron. And then we'll talk about spinal cord structure and also the reflexes, and we'll talk about two examples of reflexes, monosynaptic and polysynaptic reflexes. And then we'll discuss about sensory pathways, the pathways through which, let's say, a pinprick or maybe different kinds of sensations, how is it being carried to the brain and where is it being processed, those senses. And then we'll move on to discuss about the autonomic nervous system, and we'll look at the differences between the two divisions of the autonomic nervous system, between the sympathetic and the parasympathetic divisions, and we'll see how they are differently regulated, what are the neurotransmitters released, and we see that the autonomic nervous system is going to be regulated through the release of various hormones, acetylcholine versus epinephrine, at the target organs, and when those hormones bind to the organs, it's going to activate G protein coupled receptors. And now I will give you a little more detail about these G protein coupled receptors, and there will be different pathways which are activated. You can have the G alpha S, G alpha Q, and G alpha I pathway. So in order for each pathway to be activated, there are different subunits released from this complex, and it really depends on which type of receptor is being bound by the hormone. And for the scope of our course, I will not tell you what are the names of the receptors. All you need to know is when a different type of receptor is bound, it could have either G, alpha S, I, or Q being released, and how these in turn will have an impact on the secondary messenger levels. So with that, let's begin with what is EPSP and what is IPSP. So EPSP stands for excitatory postsynaptic potential. IPSP stands for inhibitory postsynaptic potential. And so the question is, what do we mean by a postsynaptic potential? And that's described right here. So postsynaptic potentials are graded potentials which developed in the postsynaptic membrane in response to a neurotransmitter. So let me explain this sentence in the picture format. So here's a picture showing you a synapse, that's the junction between these two neurons, and on top you have the presynaptic neuron, and then below that you have the postsynaptic neuron. So the presynaptic neuron, when an action potential travels down it, it's going to release these vesicles containing the neurotransmitters, and these neurotransmitters can bind to channel receptors on the postsynaptic membrane, and that can lead to a graded potential. Now these graded potentials can be divided as either being excitatory or inhibitory. And in an excitatory postsynaptic potential, there would be a depolarization of the membrane potential, and what we mean by depolarization is that the inside of the cell gets less negative. And it's called inhibitory if you have a hyperpolarization of the membrane potential, and hyperpolarization is where the inside of the cell gets more negative. Now the question is, what can lead to either depolarization or the hyperpolarization of the membrane potential? So if there are the opening of chemically gated sodium channels, sodium ions will flow into the cell, and it can make it less negative on the inside, and that leads to the depolarization of the membrane potential. On the other hand, if you have the opening of either chloride channels, which means negatively charged chloride ions enter in, or the opening of potassium channels, where the positively charged potassium ions leave the cell, that will cause the inside of the cell to get more negative, and that is hyperpolarization. Right? So we see that in the case of inhibitory postsynaptic potential, there would be a hyperpolarization, and in the case of the excitatory postsynaptic potential, there would be a depolarization. And all of the changes, whether it is hyperpolarization or the depolarization, that is going to be integrated by this region, which is the axon hillock. And if the threshold is reached, an action potential will be generated and propagated, right? And what do we mean by the threshold? It's the membrane potential of between -60 to -55 millivolts. That's the threshold. And if that threshold is reached, the action potential is generated. And if you have a hyperpolarization, that means the threshold won't be reached. It will become more negative on the inside, and there won't be any action potential being generated. Did you have a question?
I was just going to ask you about EPSP. Is that always happening during the depolarization?
Yes. So it's the postsynaptic potential. It could, if it's causing depolarization, it would be excitatory, because that would eventually cause the threshold to be reached. All right. So this type of regulation not only happens in the postsynaptic neuron, but it can even happen in the presynaptic neuron as well. Right. This type of regulation can also happen in the presynaptic neuron, as we will consider in this example. So in this figure, what you see is an axonic synapse. So you find that there's this yellow color neuron that has an axon here, and then you have a green color neuron. You see the axon here as well, and they are synapsing onto the input region of a purple neuron here. And so we'll see how this axonic synapse can lead to synaptic inhibition or presynaptic facilitation. So in the example of presynaptic inhibition, what you notice is that in this green neuron, as the action potential comes down, it's going to cause the release of a neurotransmitter known as GABA. Okay, and what GABA does is it's going to inactivate the calcium channels in this yellow colored neuron. So it's going to inactivate the number of calcium channels, and notice here that out of the 4 calcium channels, 3 have been inactivated by GABA, and only 1 calcium channel is shown in this cartoon, which is still active. And so when this yellow color axon is going to get depolarized due to the action potential, that will cause fewer calcium ions entering into the cell. And I told you that in the output region you have voltage gated calcium channels which permit calcium to come in. Notice that fewer calcium ions enter, and as a result you will have fewer vesicles fusing with the cell membrane, right? And we know that you need calcium for the SNAR complex to be activated, for the vesicles to fuse with the membrane. When you have less calcium, there are fewer vesicles fusing with the membrane, and as a result you have less neurotransmitter released, and as a result there will be a reduced effect on this postsynaptic neuron. So this is how there is presynaptic inhibition. Now the flip side of this could be by having a different kind of neurotransmitter released, which will not inactivate calcium channels but in turn activate calcium channels, and that is shown in this right-handed figure. So the action potential is arriving down the green axon, and that leads to the release of serotonin, right? This is a different type of neurotransmitter, and serotonin will activate these voltage gated calcium channels. And so when the action potential travels down the yellow color axon, that will cause more calcium to enter into the cell. And when you have more calcium ions, you will have more of the SNAR complexes in the vesicles being activated. That leads to more vesicles fusing with the cell membrane, releasing more neurotransmitters, and as a result there will be an increased effect on the postsynaptic membrane. Alright, so we see that this type of regulation happens presynaptically and also postsynaptically as well. Are there any questions? Yes?
What about the action potential influences what neurotransmitter gets released?
That's beyond the scope of what we're discussing. Right? Yes?
So on the yellow axon, the action potential there is going to control the neurotransmitters being released?
Yes. So when the action potential reaches this the terminal end of the axon, that's going to activate voltage gated calcium channels, right, in this yellow neuron, but here it's going to cause different types of neurotransmitters to be released. Yes?
Does the GABA
Yeah, so GABA is eventually downstream. It will prevent it from reaching threshold because you have fewer ligands being released from the yellow neuron. What was
Does
that?
it take more time to get to threshold, or
It really, see, it's beyond the scope, right, on which neurons are going to release which type of neurotransmitter. But I'm just saying that you have this regulation happening in the postsynaptic neuron and in the presynaptic neurons as well. That's all I want the take-home message to be from this slide. Okay, so I have a question here to help you better understand what you need to take away from this. So if you have the binding of a ligand in the postsynaptic terminal, which is going to cause the influx of chloride ions, then that will result in what? Will it result in an action potential, in EPSP, IPSP, or in depolarization? What do you think would happen if you have the influx of chloride ions? TS can help you. Okay, we have lots of responses. Ronan, do you have anybody in your group who wants to volunteer an answer? What is it, guys? A, B, C, D, or E? What is it?
I chose C because it will create hyperpolarization.
You chose B?
C.
Oh, C, okay. Most of you chose C, and that is the right answer, right? So we see when chloride ions come in, it will cause a hyperpolarization of the membrane potential, and that would result in the inhibitory postsynaptic potential because negatively charged chloride ions enter into the cell. Is that clear, folks? Yes? Why it is IPSP caused by incoming chloride ions? Yes? Okay, all right. Any questions at all? Okay, if there are no questions, we can switch gears and talk about the structure of the spinal cord, and I'll introduce you to what is known as the dermatome. Alright, so what you see here is the posterior view of the spinal cord, meaning the view from the back, and we see that the spinal cord has got 31 spinal nerves, right, and it's been divided into different regions. You have these cervical spinal nerves, which are colored here in blue. Following that, you have the thoracic spinal nerves, and then the lumbar spinal nerves, sacral, and the coccygeal spinal nerves. I'm not going to ask you to regurgitate all these names, right? But the question you may be wondering is, why are they numbered in this manner? Why do we have C1, C2, and then T4, T5? Why don't we just all have them numbered from 1 to 31, right? And that's a valid question. And if you were to take a cross-sectional view of the spinal cord, we see that the arrangement of the white and the gray matter is going to be very different between this cervical region versus the thoracic, lumbar, and also the sacral regions. There will be a very different arrangement in the gray and white matter. You all know that gray matter contains the neuronal cell bodies, and the white matter will carry the axons, which will have the axons which carry information to and from the spinal cord. And so while the functional significance of this, we can't get into its detail, just know that the way in which the input signals and the output signals are being processed will be unique in each of these regions. We also see that each of these pairs of spinal nerves, they're going to innervate the surface of the body, and this is known as the dermatome. So we see that each pair of spinal nerve is going to monitor a surface in the body, and this entire mapping is known as a dermatome. Now, the clinical significance of having this dermatome map is that if there is an injury in any part of the spinal cord, you could identify that by considering which part in this dermatome is affected. For example, let's say a person is not able to feel his thumb, right? It's lost sensation there. Then it's likely possible that maybe there's something affecting the C5 nerve, right, on that side of the body, and that's why maybe that thumb has lost its sensation. We also see this dermatome has clinical significance when we consider the pathology known as shingles, right? So shingles is a painful, it's a condition where you have these painful rashes occurring along certain parts of the dermatome. And this is caused by the chickenpox virus, which may be dormant after a person has been affected by chickenpox. These viruses are dormant in the spinal nerves, and something could reactivate it. It could be stress, maybe that person is immune compromised, so that could reactivate these viruses. And depending on where the virus was residing, we'll see that this rashes will occur on that part of the dermatome. So by seeing this characteristic rash pattern appearing along that part of the dermatome, we could determine which spinal nerve was affected. Right, so that's just a couple of clinical applications for the importance of the dermatome and how the spinal nerves innervate these various regions. When you also think about the spinal cord, I want to highlight that it's protected by these layers known as the meninges, and the meninges has got 3 layers, right? So collectively it's known as the meninges. The outermost layer is known as the dura mater. Deeper to that you have the arachnoid mater, and then the deepest layer is the pia mater. So these 3 layers will be found not only in the spinal cord, but also you will find them in the brain as well, offering protection. And the purpose is to provide physical stability, and it can help to absorb shock as well. Now, based on the layers in the meninges, I want to talk about a couple of clinical applications as well. So a cross-sectional view of the spinal cord reveals that there is a space between the dura mater and the vertebral column. There's a space, and the space is known as the epidural space, because epi means above, and because it's above the dura mater, it's known as the epidural space. So how many of you have heard the term epidural injections? Anybody? Yes. All right. So this is a injection which is offered especially during labor and delivery of the baby to help with the pain management. And so the analgesic would be injected into the epidural space, and it can help target only specific nerves in the body, and it can numb certain parts of the body. So during labor and delivery, the epidural injection will help just the lower part of the body to be numbed, so the mother can still be communicating and awake during the delivery, but just the lower part of the body, there will be no sensation, and that can help with pain management. The second example I want to point out is the subarachnoid space, right? So this is the space between the arachnoid mater and the pia mater. So sub means below, and this is a space which is below the arachnoid mater. And here's where you'll find the cerebrospinal fluid being circulated. We talked about the cerebrospinal fluid being produced by the ependymal cells in the central nervous system, but it is in the subarachnoid space where cerebrospinal fluid can be collected during a lumbar puncture or a spinal tap. And this is something which is done to use the cerebrospinal fluid as a diagnostic material, right, to see if there's any infection such as meningitis and so on. So this is just pointing out how the names of these layers protecting the spinal cord, how knowing that will help you understand a couple of clinical applications as well. So with that, I want to point out the anatomical and functional organization of the spinal cord, and this is important in order to understand how reflexes function. So we see the left-handed side, there is nothing labeled because I want to point out some anatomical landmarks. So you see that the gray matter is in the middle, right, and surrounding that you have the white matter, and the gray matter is where you have the neuronal cell bodies, and this is where you will have the information being integrated and also commands being initiated. So input can come in, can be integrated, and appropriate responses can be initiated from the gray matter. Whereas the white matter you will have is made up of all the axons, which is carrying the information, and so this will carry the input signals and also carry the output signals as well. And we find that the orientation of what you're seeing here is that the subject is lying face down with the feet facing towards you and the head towards the board, right? And so in that manner of orientation, this here would be the posterior horn, and down here we would have the anterior horn, right, in the spinal cord. And we find that in the posterior horn there are two functional divisions. You have the sensory nuclei, the posterior horn, and the anterior horn you have the motor nuclei. And I mentioned previously, nuclei in the central nervous system is just referring to the collection of the neuronal cell bodies, right? Whereas collection of neuronal cell bodies in the peripheral nervous system, it's known as the ganglia, right? So this is just showing you where the neuronal cell bodies are. And so sensory input is being brought to the sensory nuclei through the posterior root, right? So the posterior root is going to bring information to the sensory nuclei, and once the information has been processed, motor output signals can be sent from the motor nuclei through the anterior root, right? So that's how information comes to the spinal cord, and commands are sent from the spinal cord as well. And having explained the anatomical and the functional organization of the spinal cord, we can have a better understanding of how reflexes work. So what are reflexes? So reflexes are rapid and automatic responses to the specific stimuli. And I want to put emphasis on the word automatic because when you have a reflex, you're really not thinking about it. It just happens automatically without you putting much any thought into it. And for a specific reflex, it's important to note that the response is going to be the same response time and again, right? So you can always expect a same type of response for a given reflex. And so to understand how the reflex works, we need to consider what is the reflex arc and also the various components in the reflex arc. So the reflex arc is just referring to the wiring, or it's referring to the ways in which the various neurons which bring in information and carry out commands, how they are connected one to another. So in the reflex arc there are 5 components. The first one is going to be the sensory receptor, right? And we talked about different kinds of sensory receptors. In this example it's showing a painful stimulus, so the sensory receptor could be a free nerve ending. And this sensory receptor is going to send signals to a sensory neuron shown here in red, and this sensory neuron will carry the signals to the central nervous system. In this case it's carrying it to the spinal cord through the posterior root. And then in the spinal cord there could be some processing done. Notice that signals can even be sent to the brain, but eventually the spinal cord is going to cause the motor neuron, which is the 4th component, to be activated, and that will send signals through the anterior root shown here, and the motor neuron can activate a specific muscle which will contract or relax, and in this case the contraction of this bicep muscle leads to the hand being withdrawn, right? So these are the various components of the reflex arc. Now let's look at the events which take place. So we see that the arrival of the stimulus, which could be the pain to the pain stimuli in the free nerve endings, it's going to activate this sensory neuron, and this will carry information to the central nervous system, and from there commands from the spinal cord is going to be carried through the motor neuron, and this arrow here is showing that some signals can even be sent to the brain in some cases. And we find that the activation of this motor neuron will cause the muscle to contract, and that's how the reflex takes place. So these are the overall steps in terms of how a reflex occurs. Now there are different types of reflexes. You have some reflexes based on development. Some are innate reflexes, others are learned through habitual repetition, like what musicians and athletes would develop, right? Those are reflexes which differ based on their development. But we're going to talk about the classification of reflexes based on their complexity. And we're going to look at two types of reflexes. The first one is the monosynaptic reflex, and the second is the polysynaptic reflex. An example of the monosynaptic reflex is the stretch reflex or the patellar tendon reflex, which you would have experienced if you go to the doctor's office, or even if you haven't and you watched cartoons, you will see the patellar tendon stretch reflex being over exaggerated. All right, so as the name indicates, in this reflex there's only a single synapse. So the sensory neuron which brings the input signal is going to directly synapse with the motor neuron shown here in black. And because there's only a single synapse, the response is very fast. Now in contrast to this, in the polysynaptic reflex, we see that it's going to be a lot more complicated, and there will be more than one synapse, as the name indicates. So the sensory neuron shown here in red is going to bring signals to an interneuron, and this interneuron will in turn pass on signals to the motor neuron. And the question is, what is an interneuron? This is the neuron which will interpret the incoming information, and it can coordinate an appropriate response. And because there are multiple synapses, this will take a longer time for the response, because it just takes more time to process the information through multiple synapses. So having this introduction, let's take a closer look at what happens in this stretch reflex versus the withdrawal reflex. So in the stretch reflex or the patellar tendon reflex, we see the stimuli is going to be tapping on the patellar tendon with the reflex hammer, right? So once this tendon is tapped on, that's going to cause the stretching of this tendon, which will in turn pull on the quadricep muscle. And in this quadricep muscle, there is a receptor known as a muscle spindle, and this is going to be activated by the distortion. And that will cause signals to be sent through the sensory neuron shown here in red, and those signals are sent to the spinal cord, and the sensory neuron synapses with the motor neuron shown here in black, and that motor neuron will be activated, which will in turn send signals to the muscle, and that muscle is going to contract, right? And this is the contraction of the muscle which causes the leg to kick forward, right? So this is how the stretch reflex works. You just have a single synapse, and the stimuli is the stretching of the tendon, and the response is the contraction of that specific muscle. Any questions on monosynaptic reflex? Okay, now let's take a look at the polysynaptic reflex, and the example is a withdrawal reflex. So here we have a painful stimuli or maybe pressure, and we see that the goal is to withdraw away from that painful stimuli. And so notice here that in this reflex you have multiple muscles being affected, right? So you have a flexing muscle, which is the biceps, that's going to contract, whereas the extensor, the tricep muscle, that's going to relax when you have the withdrawal reflex. And the strength of the response is going to depend upon the strength of the stimuli and also the location as well. So let's see how this takes place. So the painful stimuli is going to activate the sensory neuron shown here in red. This is going to send signals all the way to the excitatory interneuron, right? And this interneuron has got multiple branches as shown here. So one branch is going to activate the motor neuron, which innervates the bicep, which is the flexor muscle, and so the bicep is going to be stimulated to flex. Simultaneously, you also have an inhibitory interneuron which is going to be activated, and that will inhibit the motor neurons innervating the tricep muscle. So while the bicep is stimulated to flex, the extensor, the tricep, is stimulated to relax. Okay, and as a result, we see that the withdrawal reflex takes place. But notice here that there is also some signals which are eventually sent to the spinal cord. So even though your hand is withdrawing very quickly, but your brain will get signals that there is a painful stimuli which it is moving away from as well. But this spinal reflex is happening without the brain sending a signal. It's just happening because of signals sent from the spinal cord. But now we're going to switch gears and talk about how does the brain detect different kinds of touch sensation. I told you there are different kinds of 5 senses. We'll only spend a little more time focusing on the touch sensation, and I want to tell you how are the signals carried to the brain and processed. So the way the signals are carried to the brain is through the first order, second order, and the third order neurons. And so we see that the first order neurons, they are also known as the primary sensory afferents. I told you afferent means it's to bring the signal, whereas efferent is to carry away the signals. And this is shown here in green for the sake of just color coordination. And this first order neuron is going to deliver sensation to the central nervous system, and in this case it's bringing signals to the spinal cord. And this has got the cell body in the posterior root ganglion, right? And I told you where the posterior root ganglion is found in the spinal cord. So that's where the cell body is. And we find that this first order neuron is going to synapse with the second order neuron shown here in blue. And the second order neuron will take the signals from the first order neuron. And I want to point out what happens to the axons which come from this second order neuron. It's going to cross over to the opposite side of the body, right? And so this crossing over is known as decussation, and I will use the term decussation and crossing over interchangeably. And then it's going to ascend all the way to the thalamus, right? And thalamus is a region found in the brain. It's going to be found deep inside it. And from thalamus, it's the second order neuron will synapse with the third order neuron shown here in orange, and this third order neuron will take the signals all the way to the primary somatosensory cortex. So we talked about the primary somatosensory cortex when I discussed the different parts of the brain. So you have the parietal lobe, which is separated from the frontal lobe and by the central sulcus, and the first gyrus on the parietal lobe would be the primary somatosensory cortex. And the signals which are sent here, that will allow it to reach our awareness. So that is how a specific sensation can be sent to the brain. Now there are different pathways. There are 2 pathways is what I'm going to mention. There are others too. I just want to mention 2 pathways by which different kinds of touch sensation can be taken to the brain. Right? So on the left hand side we have the spinothalamic pathway, and on the right side we have the posterior column medial lemniscus pathway as well. So I will abbreviate this as PCML. Sometimes it's also called as DCML because the term posterior and dorsal are interchangeable. Okay, and so this spinothalamic pathway, its goal is to carry crude touch sensation. Examples of crude touch sensation would be maybe light pressure, pain, changes in temperature. Those are things which the spinothalamic pathway is going to carry. On the other hand, this PCML pathway is going to carry fine touch sensation. So examples of fine touch sensation would be vibration, deep pressure, and even proprioception. So proprioception is just a sense of knowing your position of the body in space, right? And if you are a person who likes to run and you often twist your ankles, probably because the proprioception isn't accurate, right? So that may be affected. So these are things which signals which are sent through the PCML pathway. Now, these are the two differences. I want to point out the similarities between these two pathways and a second difference as well. Right? So, we see that the first order neuron is going to bring signals to the spinal cord, and in the spinal cord it's going to synapse with the second order neuron. And notice that the second order neuron, it's going to decussate immediately, right? It moves over to the other side, and then it's going to ascend all the way to the thalamus, shown here in blue, and that second order neuron is going to synapse the third order neuron, which will in turn take signals to the primary somatosensory cortex. And notice that the different parts of the primary somatosensory cortex, where signals from the tongue are processed, from the jaw, from the lips, from the face, upper limb, from the torso, lower limb, and even from the genitalia. So there are different regions in the primary somatosensory cortex where these signals from different parts of the body can be processed. Now let's compare the spinothalamic pathway to the PCML pathway. So again here, the first order neuron is going to bring signals to the spinal cord, and notice that instead of synapsing with the second order neuron in the spinal cord, it's going to ascend all the way to the brainstem. Right? Specifically, the region in the brainstem to which it's going to ascend to is the medulla oblongata. Right? So it's the medulla oblongata where it reaches, and at the medulla oblongata it's going to synapse with the second order neuron. And the second order neuron, just as before, it's going to decussate immediately, and it's going to ascend all the way to the thalamus through the medial lemniscus tract, right? And I hope you understand how the name posterior column medial lemniscus came about. So you may be wondering, what does lemniscus mean? It just means that ribbon is the meaning of the word lemniscus, and because this pathway here looks like a ribbon, anatomists named it like that. All right. So it goes and synapses with the third order neuron in the thalamus. From there it takes the signals to the primary somatosensory cortex. So let me just tell you the differences between spinothalamic and the PCML pathway. Spinothalamic pathway will help to detect crude touch sensation, whereas the PCML will help detect fine touch sensation. And here in the spinothalamic pathway, the decussation happens down in the spinal cord, whereas in PCML, the decussation takes place all the way in the brainstem, specifically in the medulla oblongata. So the clinical relevance of the decussations of these two pathways at different regions in the central nervous system can be understood in the medical condition known as Brown-Séquard syndrome. Right, so this is a condition where there is only a part of the spinal cord being affected. Right, so the lesion to this one part of the spinal cord could be due to maybe as a tumor, or maybe there is trauma, or maybe there could even be inflammation. So something is causing only a part of the spinal cord to be affected. So when this happens, there is no surprise that the side where the spinal cord is affected, motor function would be impacted, right? But on both sides of the body, some type of sensation will be affected as well, right? So we see that on the opposite side of the body there will be a loss of crude touch sensation. So things like pain, temperature, light touch can be detected on the opposite side of the body, whereas on the same side of the body, in addition to loss of motor function, there will also be loss of the fine touch sensation, which includes proprioception, vibration, and even deep pressure as well. Right? So I hope you understand why only on one side one type of sensation is affected, because you have the spinothalamic and the PCML pathways carrying the various types of touch sensation. So this figure here is just showing you the PCML pathway drawn in red, and then you have the spinothalamic pathway drawn in green. And the question would be, if there is, let's say, the right side of the spinal cord being damaged, how would it affect the crude and fine touch sensation on the left and right side? So the quick way I like to remember this is that on the same side you will have motor function affected and also fine touch sensation affected. And on the opposite side, you have the crude touch sensation being affected. So here's a sample question which I want you to think about, right? So damage to the left side of the spinal cord above the level of the legs would cause which of the clinical symptoms? Would there be loss of pain sensation in the left leg, or would there be loss of proprioception on the right leg? Would there be loss of fine touch in both legs? Would there be loss of temperature sensation in the right leg? Pretty easy question, right? I'm kidding. It's probably one of the most challenging questions in this entire course. So you can talk to your neighbors and turn to your TAs and see what you guys think. I'll give you a minute and then we'll move on. I just don't have poll everywhere for this because it's better to hear an answer from anyone who gets it.
Right side. Up. Yeah. Good. Yes.
No, there's no poll everywhere for this. Okay, does anyone have an answer? Yes? Yeah, anybody can answer, yeah. D, okay, wow. B is the right answer, alright? And I'll let you figure that out. Okay, let me tell you, I won't be mean. So we see that temperature sensation is an example of crude touch, right? And that's going to be affected on the opposite side of the body. And so that's why crude touch being affected on the opposite side means on the right side of the, on the right leg, you won't have temperature sensation. Okay, excellent. So I'll give you an example, and in the exam we'll have a similar question, maybe be a bit easier, but this is a concept which I want you to come prepared for. Okay, moving on, I want to talk to you about the sensory and the motor homunculus, right? So this sensory and motor homunculus, they're just representing a map of the brain areas which are dedicated to sensory and motor processing for different anatomical divisions, right? So it's a map of the brain areas for sensory and motor processing and how it's devoted to different anatomical regions. So we see that the motor cortex here is shown in red. I notice that there are different parts of the body where the senses are being, where there can be signals sent for motor control, like the mouth, the hands, legs, and so on. And then the somatosensory cortex is shown here in blue. I notice that sensory input from different parts of the body, they are being processed in different regions. So if you were to take all of these devoted regions and then make it into a human structure, that's what this would look like. So you see that the hands are really large, meaning that you have a lot of sensory neurons innervating the hands, the lips, and the tongue. And so the sensory homunculus is a functional map of the somatosensory cortex. And we see that the reason why the hands and the mouth and the tongue it's much larger is because it is showing you it's proportional to the density of the sensory neurons innervating those regions, and it's not proportional to the size of that organ. So if you consider here, you see that the genitalia have a much smaller region devoted, and that's why it's shown in a very small way. So similarly, we find that in the motor homunculus, this is the functional map of the primary motor cortex, and you see that the fine motor control is possible by the hands, the mouth, and the tongue. But you notice that there is no genitalia represented here because genitalia cannot be moved. And so the hands, face, and tongue, they appear a lot larger, but the trunk is relatively smaller as well. Are there any questions? If there are no questions, I'm going to quickly talk about the autonomic nervous system. So this system is responsible for involuntary control, right? And we see that signals can be sent from the brain and spinal cord got to various target organs, and this will affect multiple organ systems. Things like the urinary system, the respiratory system, digestive system, cardiovascular system, all this will be controlled by the autonomic nervous system. And this will help to adjust the composition of body fluids, such as the water levels or maybe the amount of dissolved gases, electrolytes, and so on. And there are two main subdivisions. We have the sympathetic division, which will regulate the fight or flight response, and the parasympathetic division, which will regulate the rest and digest response. And whenever we talk about this, I like to tell people about something which happened to me a few years ago. So I was driving down the road from Cleveland to Ithaca, and I was very drowsy, and all of a sudden I saw on the right hand side there was a deer standing, and as I got closer I saw on the left hand side there was another deer, and I went right in between them, right? And so what coffee couldn't accomplish for the last couple of hours, those deer accomplished in a moment, right? And there was no doubt about it, I had a heightened sense of mental alertness during the sympathetic system being activated. There was increased metabolic rate, definitely had decreased digestive and urinary function, increased respiratory rate, of course, dilation of the respiratory passageways, increased heart rate, blood pressure, activation of energy reserves, and of course activation of sweat glands as well. So many of these things we'll discuss about when we talk about the different organ systems in the upcoming lectures. And so a couple of years ago, this picture was circulated. Anybody recognize this? Not the TAs. Yes, there was a bear on campus, right? So if you were one of those who happened to see the bear on campus, you probably would have sympathetic activity being increased. Now the opposite to this would be that response of the parasympathetic activity, which would be decreased metabolic rate, reduced heart rate, probably most of you have this right now, increased secretion of salivary glands and digestive glands. This is something we'll talk about in the digestive system. Increased motility of the digestive tract, and also increased blood flow in the digestive tract, so nutrients can be taken and absorbed and taken to the various parts of the body. And also urination and defecation will also be stimulated as well. So these are some general responses which we'll cover in greater detail in the specific organ systems. But what I want to do here is point out the neural pathways and how they differ between the sympathetic and the parasympathetic division. So we see that the preganglionic neurons, they're going to arise from the central nervous system, and they're going to be a lot shorter in the sympathetic division. Whereas in the parasympathetic division, you see these neurons, they're much longer. And these preganglionic neurons will both release acetylcholine, and acetylcholine will bind to ligand-gated ion channels, right? And so they release these acetylcholine into the postganglionic neurons. Now these postganglionic neurons in the sympathetic division, they're going to be a lot farther from the target organs. So notice the target organ is here, and so you could have these really long axons going to the target organs, or these postganglionic fibers, they can just release their neurotransmitters or hormones into the bloodstream. So we find that in the postganglionic neurons in the parasympathetic division, they are much closer to the target organs. So in the case of sympathetic division, we would have epinephrine and norepinephrine. They will be released into the bloodstream, or they can be directly released to the target organs as well. Whereas in the case of the parasympathetic division, we see that these fibers are much shorter than the sympathetic division because they are closer to the target organs, and they will release acetylcholine to G protein coupled receptors, right? So even here epinephrine and norepinephrine will bind to the G protein coupled receptors. So in the last minute, let me just point out what this looks like when you look at the actual central nervous system and the various organs which are innervated. So you see the red lines indicate the preganglionic neurons and the respective fibers, whereas the black lines will indicate the postganglionic neurons. And these preganglionic neurons are all going to be in the spinal cord for the sympathetic division, whereas in the parasympathetic division, some of them are in the brainstem, others are in the spinal cord. We also see that the nerve fibers are going to be shorter in the sympathetic division compared to the parasympathetic division, where they're going to be much longer. And the postganglionic neurons, they will be further away from the target tissue, whereas in the parasympathetic they're going to be a lot closer to the target tissue. And we see that there is greater divergence in the sympathetic division. So if you have sympathetic activation, multiple organ systems can be activated simultaneously, like you have increased heart rate, increased blood pressure, whereas here you have fewer systems being activated. And then we find that the main difference is that acetylcholine is released to the target organs in parasympathetic division, whereas epinephrine and norepinephrine are released in the sympathetic division. And we will talk about dual innervation when we come back tomorrow, and that will lead us to the discussion on autonomic tone. Thank you all for your time.